Welcome viewers. Today I will discuss on vaccines. I will tell you the mechanism of vaccination in brief, different types and natures of vaccines and advantages and disadvantages of different vaccines. Now vaccine is a biological preparation which protects us from a disease. Vaccine contains an agent which resembles an organism or part of it, and when taken it activates the immune system. Now I will tell you the mechanism of vaccination. The agent present in the vaccine when enters into the system, the antigen presenting cells present in there like macrophages, dendritic cells, they engulf the antigen, they process it and they present short fragments on their surface. Now T lymphocytes recognize these fragments as foreign and they are activated, both T helper lymphocytes and TC or cytotoxic T lymphocytes. Now these T lymphocytes after activation, some of them are converted to effector T lymphocytes and some of them are converted to memory T lymphocytes. At the same time, the B lymphocytes are also activated by the same agent. B lymphocytes are activated, some of them are converted to effector B lymphocytes or plasma cells which secrete immunoglobulins, and some of them are converted to memory B lymphocytes. These memory T lymphocytes and B lymphocytes provide protection in future against the same agent if it enters into the system. This is the mechanism of immunization. Immunization can be achieved actively or passively. The active immunization can be acquired naturally through natural infections or artificially through vaccination. And passive immunization can be acquired naturally and artificially. The best example of naturally acquired passive immunity is transfer of immunoglobulins from mother to the fetus through the placenta and transfer of immunoglobulins from mother to the baby through breast milk. And the example of artificially acquired passive immunity is when a patient is in emergency after snake bite or tetanus intoxication, preformed antibodies are given to him. The induction of immunity through vaccination was developed by the Chinese and Turks in 15th century. And this technique was brought to England by Lady Mary Montagu in early 18th century, who was a wife of a British ambassador and went to Turkey in early 18th century. She observed the technique and brought it to England and saved the lives of many people. Later in 18th century, Edward Jenner discovered a safer technique for vaccination. Edward Jenner noticed that milkmaids who had suffered from cowpox would never catch the smallpox disease. And he reasoned that if fluids from the cowpox lesions taken and inoculated in an individual, he might be resistant to smallpox. So he deliberately took the cowpox fluid and injected it into an 8-year-old boy, and after 6 weeks again he infected the boy with smallpox virus. And as predicted, the boy did not catch smallpox. He named this practice or technique vaccination. The Latin word vacca means cow. Since this resistance came from cowpox, he called it vaccination. And this technique then spread throughout Europe very quickly, and as a result in 1980 World Health Organization declared the world and its peoples free from endemic smallpox. Later Louis Pasteur elaborated the concept of Edward Jenner, and Louis Pasteur developed vaccines against rabies, anthrax and many other diseases. Now there are 3 types of vaccines have been developed so far. The very early vaccines are called first generation vaccines and they are composed of whole organisms. The second generation vaccines are composed of purified macromolecules from the organisms, and third generation vaccines are developed using recombinant DNA technology. where directly the genetic material is used as a vaccinating agent. The genetic material enters into the system, it is expressed into a protein. That protein is considered as foreign by the immune system. Immune system activates and generates immunity against that foreign protein. I will tell you mainly first generation vaccines and second generation vaccines. First generation vaccines are of two types, live attenuated vaccines and inactivated or killed vaccines. Live attenuated vaccines contain whole organism but in attenuated form. Attenuated means reduction of the pathogenicity of that organism. Attenuation can be achieved by growing the organism under an abnormal condition for a long period of time. The live attenuated poliovirus which is used today is called Sabin vaccine after the name of its discoverer Albert Sabin. It is composed of 3 attenuated live poliovirus strains. And when given orally, these 3 strains colonize in the intestine and they activate the immune system. And several doses are required for this process. Now, what are the advantages of live attenuated vaccine? Since the organisms are growing inside our system, a single dose is sufficient for live attenuated vaccines. And they generate very strong immunity. Also, both cell-mediated and acquired immunity is generated in case of live attenuated vaccines. And what are the disadvantages of this type of vaccine? The great disadvantage is the live attenuated organism can revert to its virulent form. The reversion rate of Sabin vaccine is 1 in 2.4 million doses. Another disadvantage is, since this vaccine is composed of live organisms, proper storage and transportation is a must for this type of vaccine. Now I will tell you inactivated or killed vaccine. Inactivation is done mainly by heat or chemical treatment. So the organism could not replicate inside the host system, but it can generate the immune system. And the killed inactivated vaccine of poliovirus which is used today is called Salk vaccine. It was discovered by scientist Jonas Salk and it contains poliovirus inactivated by formaldehyde. This vaccine, inactivated vaccine, has several advantages over the live attenuated vaccines. Like these vaccines are safer and the constituents are clearly known and they could not cause the disease. What are the disadvantages of this vaccine? Since this vaccine contains killed microorganisms and they don't replicate inside our system, several doses are required for this vaccine. And this vaccine mainly induces the humoral immunity, not the cell-mediated immunity. And if killing is not proper, some of the viruses remain alive, then it may cause the disease. To minimize these disadvantages, second generation vaccines came into the market where instead of whole organisms, only part of it is used, like toxins produced from an organism or glycoproteins present on the bacterial or viral surface or polysaccharides present in the bacterial capsule can be used as a vaccinating agent. There are two types of subunit vaccines, toxoid and vaccines where bacterial polysaccharides have been used. Several organisms produce exotoxins which may cause a disease. So, if these toxins are inactivated by some way and then they are used as vaccinating agent, they may activate the immune system and provide protection against the particular organism. Like tetanus, diphtheria, these toxins are chemically inactivated and after chemical inactivation, these toxins are called toxoids. These toxoids are used as vaccinating agents. Immune system develops antitoxoid antibodies and these antibodies provide protection in future. Similarly, sometimes some bacteria have a capsule full of polysaccharides, and presence of polysaccharide prevents the immune system to look inside it, and the bacterium escapes from the immune system. So, if these polysaccharides are isolated and used as a vaccinating agent, then it may activate the immune system, then immune system can develop antibodies against the polysaccharides. Currently available Streptococcus pneumoniae vaccine contains 23 different capsular polysaccharides in it. If these small molecules like polysaccharides or glycoproteins are conjugated with a large carrier protein, then it would be better for immunization, because immune system will look into a big molecule and it will be activated more prominently. This is the concept of conjugate vaccine. The Haemophilus influenza type B vaccine contains type B polysaccharide conjugated with the tetanus toxoid. So, when taken inside, the immune system recognizes it and it generates antibodies against the type B polysaccharide. So, when Haemophilus influenza type B enters into the system, the antibodies present, they will try to prevent the disease. Now, what are the advantages of this type of vaccine? These vaccines are safe compared to the first generation vaccines, and since immune system is activated against specific antigen, it lowers the chance of other adverse effects. And the disadvantage is the development of immunity is difficult in case of second generation vaccines. Now, sometimes vaccines are combined and given to the patient. It reduces both the number of clinic visit and number of injections. Now, I will discuss how vaccines are produced. There are several stages for vaccine production. First step involves identification of the antigen. If it is a virus, then we have to grow the virus in living cells. Mainly for a virus, the embryonated eggs are used. Sometimes specific cell types which can divide repeatedly, they are also chosen. For a bacterium, bioreactor is used. Next step involves purification and isolation of the antigen. The virus or the bacterium is isolated from the growth medium and it is purified. Next step involves modification of the organism. If it is a whole attenuated vaccine, then the organism must be attenuated by some means, and for other vaccines, some components like preservatives or antibiotics should be added. Next step involves quality control and then packaging, labeling. And this procedure, starting from antigen selection to production of vaccine, it requires almost at least 9 months and 9 to 22 months depending on the nature of the vaccine. Now, the vaccines should be stored and preserved at low temperature, because if it is a whole organism vaccine, especially live attenuated vaccine, the vaccines should be stored properly at a low temperature, because if it is not stored properly, then failure of that vaccine may occur. Now, in vaccines, other than the active component, we will always find some other excipients like adjuvants. Adjuvants are used to enhance the immune response to a vaccine. Mainly in case of second generation vaccines, adjuvants are used. Preservatives are given to prevent fungal or bacterial contamination. Sometimes gelatin, additive like gelatin is added, which stabilizes the vaccine from adverse conditions like freeze drying or heat. Or sometimes some inactivating agents like formaldehyde or yeast protein or egg proteins are found in the vaccine, which comes from the manufacturing process. Now, vaccines can be administered by several routes, like it can be given orally or intramuscularly, subcutaneously or intradermally. There are several schemes for vaccination or immunization for babies, children, adult people. Now, some of the vaccines maintain immunity for a prolonged time, like measles, mumps, rubella, yellow fever virus. They protect for a long period of time, whereas cholera vaccines give protection for short periods. Some vaccines are 100% effective, like measles, mumps, rubella, yellow fever, whereas some vaccines are 80 to 95% effective, whereas some vaccines are 40 to 60% effective. Now, there are some common reactions observed after vaccination, like local reactions like redness, swelling at the injection site, also sometimes small nodules appear, and some general reactions like fever, nausea, diarrhea. These are also observed following vaccination. Still, vaccination is better than suffering from the disease. So today we discussed different types of vaccines, how vaccines work inside our system, what are the advantages and disadvantages of different vaccines. We learned today that first generation vaccines are better compared to the second generation vaccines, but second generation vaccines are safer compared to the first generation vaccines. How vaccines are produced in the laboratory and how they are given via different routes, how they should be preserved, and there are several immunization charts for babies, children, adult people, and we should follow those charts for proper vaccination.